Hey guys, so we are in a new chapter. Chapter 10 is biotechnology. It's one of the coolest chapters because it's talking about all the neat stuff that's happening currently and the technology that we use. So, what is biotechnology? That is going to be when we artificially modify genetic material of organisms. So, we have technically been doing this for a really long time, since basically the dawn of agriculture, when we picked certain plants and we liked those certain plants, so we grew those specifically and bred them with other ones. That's manipulating the outcome of the situation. So that's biotechnology is agriculture. The dogs that I have in my house right underneath my feet right now, those are us manipulating the traits that we like the best, that we were seeing in the dogs and the wolf, and breeding that. So we have been doing this, modifying genetics for an outcome that we wanted for a long time. More recently, when we're talking about biotechnology, usually it's modifying genetic material molecularly that we are getting in there manipulating. So we're going to get into all of that stuff later, but do know that we have been using biotech to modify genetic material, produce compounds, bioremediate, um, for a really long time. Um, Science is always evolving. Of course it is. So one of the things that we're going to talk about in this video is PCR, preliminary chain reaction. And so if you were in Labster, you got to do this. I believe so. Use a PCR maybe in the Labster. But what it is going to do is it's basically like a copy machine. It copies DNA for us, which is really cool. We're going to go through the process of it. And we wouldn't have this biotechnology if it wasn't for a guy 50 years ago. This article just came out today, from this USA Today article. It came out literally today. So I had to put it into the PowerPoint, talking about how we discovered this bacteria that was growing in this hot springs in Yellowstone. There's tons of bacteria in archaea that live in Yellowstone. It's a super cool place to visit. If you have not visited, please do. I always recommend it. Go, travel, see things. This is Yellowstone, and it has these hot springs that are filled with all sorts of really cool bacteria, but they didn't know what they did. So in the 60s, they started doing some surveys and some samples with college students helping this research project. And so they collected the samples of this bacteria. They determined all the different species, and there's an enzyme in one of these bacteria that we use in the PCR machine, and it's great because it can tolerate heat. It can tolerate heat. So pretty cool about something that was figured out in the 60s, but we didn't actually use it until like 50 years later. So it is always evolving, always. I've got links for articles in this video that are 4 years old, and who knows how long, how many modifications have been made since then. So let's get started on all this new cool stuff that's been happening in the last couple decades, in the last couple years. What is going on? So let's talk about the genetic material. Just to refresh, we have talked about this in previous chapters. We are going to just talk about it real quick. One more time, we talked about nucleic acids and that they were made out of nucleotides. What were the three parts of the nucleotides? There's our phosphate, here's our sugar, here is our nitrogenous base. If we're talking about DNA, it was deoxyribose sugar. If we're talking about RNA, it was ribose sugar, and then different nitrogenous bases. That phosphate group, it's going to have a net negative charge. Look at these phosphates. Here's our phosphate, and here, look, negative. Here's another phosphate. It's a negative charge over there. That negative charge is going to be important. It gives DNA an overall net negative charge. And when we talk about future things like gel electrophoresis, the charge of DNA matters. It's going to be pulled across through the gel because it is going to be attracted to a positive, because DNA is negative and it wants to meet up with the positive. So phosphates are going to have a net negative charge. Know DNA's physical structure, that is a double helix. Know what a genome is. It's the genetic material of an organism. We have DNA, eukaryotes have DNA, prokaryotes have DNA. If we're talking about viruses, viruses can either have DNA or RNA. They may have RNA. COVID has an RNA genome. So it just depends on what that organism is. And then we talk about RNA. What was the function of RNA? We talk about central dogma all in chapter 9 last time about DNA to RNA to proteins. We needed RNA to create the proteins. So let's talk about one of the things that we can do when we are talking about getting the genome out of something is we have to get the cells. And then once we have the cells, we need to extract the DNA from the cells. So if we did this in a lab, we would lyse the cells, we'd use a detergent, some sort of kind of detergent soap that would break apart the cell membranes. And then we would be able to get into the organelles inside of the cell. What are we looking for? If in this case, we're looking for DNA. So we're going to have enzymes, proteases are going to break apart the proteins. RNases, if it ends in an A's, it's an enzyme. RNases are going to break apart the RNA, and that's going to leave us with DNA. And here it's going to talk about how it centrifuges, it's going to spin it down, DNA is going to float to the top. And then we can use ethanol to also help that precipitate out. And we can stick it in there and grab the DNA and pull it out. If we were in a face-to-face lab, you'd actually get to do this. It's a really quick, easy lab where we harvest the DNA from strawberries. It's really neat. You can harvest your own DNA from your own cheek cells, which is really cool in this video if you want to watch it. It's really basic materials to do this. It's like dish soap and a few other things, and you can pull out your own DNA and at least look at it, which is kind of neat. So that is DNA extraction, being able to get the DNA out of the cells. And in this case, we were looking at DNA, but maybe we wanted to look at RNA. We wanted to look at what RNA is being produced, what genes are being expressed. That's the whole purpose of RNA, is to help out gene expression. So maybe that's what you were looking for, and you would use a DNase to get rid of the DNA, you'd use a protease to get rid of the protein, and then you'd keep the RNA and look for that. Or maybe you were looking at proteins. It all depends on what you want to look at. In this instance, we are looking at DNA extraction. Gel electrophoresis is something that I do think, I'm really pretty sure we did in Labster, and it is really cool. You also get to do this in face-to-face. I wish you would be able to get to do this in a face-to-face because it's just neat to do. It's neat to say that you've been able to run one of these. I think it's cool. So the way it works is you are going to have, here is your machine, and in your machine, you're going to have a tray, and that tray is going to hold your gel, the actual gel, the agarose gel. And that gel is going to have these little holes in there called wells that you have created because you put in this little comb. And when you pull the comb out, it's just like a Jello mold. You pull the comb out and you end up with these little holes in there called wells. And that's where you put your material. What material is going to go in there? Where you are going to have your DNA that we're going to put in the different wells. So with the DNA, you're going to want to not use a whole strand, right? We're not going to use every bit of the DNA. We're just going to look at certain sections of it. So it will be cut down with restriction enzymes that we're going to get to in a little bit. It's going to be pieces of DNA, not the whole strand. If it was the whole strand, this wouldn't work. So we're going to have pieces of DNA, and each little thing is going to go into these little centrifuge tubes. We'll extract them from the centrifuge tubes, and we will put them, just like it says here, in the little wells. And then remember, what was the charge of DNA? Was DNA negative or was DNA positive? DNA's charge was negative because of those phosphates on the end. So DNA is negative, and we're going to put all these DNAs here on the negative side. And then we're going to put the tray in. We're going to pour the buffer solution over top of the gel, and then we are going to hook up the battery to this kit. And so there is a negative charge over here, and there's a positive charge over here. It's just like your car battery, and it's going to turn on, and you're going to run this thing for about 15 minutes. The DNA is negatively charged. It's going to be attracted across the gel to the positive charged end. So we are going to see the DNA move through gel, and you will see banding occur. It is important to note that the smaller sections of DNA are going to move faster than the bigger sections. It's easier for a smaller molecule to wiggle its way through the gel than it is a much larger piece of DNA. So you're going to see the smaller pieces run the fastest. They're going to be the furthest from the origin. They're going to be the closest to the positive side. The bigger chunks are going to be slower, and they're going to be close to back where you started from. So where we started from, these are the longer, slower molecules, and the smaller, short guys run faster. And then you're going to have your completed gel here. And if we were doing this in a face-to-face lab, we would not use the, we wouldn't use these guys and a UV light. We are going to use, or we can use these guys and they're going to have a blue dye on it. And then when we put it on top of a light box, or really you could just stick it on top of your iPhone flashlight and you could shine the light up through it and you will see the banding pattern, which is really cool. So you'll be able to compare this banding pattern to a ladder that's going to have all the bands, and you're going to be able to match up, you know, this band that shows up here. It definitely has a reference point over here. So your ladder is your reference point, and then you have your different samples to compare them to. And so we can see, you know, this sample is slightly different than this one, it's different than this one. All of these are different, but that is how this runs. And then you just can compare. You know, if we were doing it in face-to-face class, we have a sample that we collected at the crime scene. And then we have 3 different DNA from subjects that we have taken. And then we're going to compare our subject A and subject B and subject C's DNA to the DNA found at the crime scene and see if any of those guys match up, any of those samples match up. And that is how we would do it in a face-to-face class. So gel electrophoresis, I think, is really neat. Again, you've done this online, but here's another animation to cover that material for gel electrophoresis. It's a really pretty cool process. PCR, polymerase chain reaction. The PCR is the thing that I told you is basically like a copy machine for DNA. You were at a crime scene and there's just this small bit of blood, this tiny bit of blood, and you were able to sample it because you're part of the forensics team and you were able to pick it up, get it into a little container, and that's all you have. Well, if you're trying to test that against, you know, 20 different people that you brought in to sample, you can't run that 20 different times. So what you can do is make copies of it. So bring in that little bitty sample and you run it through the PCR and it makes copies, 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 copies. That's what the PCR machine does. So the steps of PCR is going to be where it heats it up really hot. And we said, when we talked about proteins, we talked about proteins when they got really hot, they would denature and they would change shape. So that's the first step in the PCR machine. It's going to get really hot, 95 degrees Celsius, and it's going to denature. And then we are going to anneal, and at the annealing stage, as we cool back down a little bit, we're going to stick on our primers. We're going to stick on our primers, and then just like we talked about in DNA replication and creating our messenger RNA, we're going to have that kind of primer where our polymerase sticks on. So our DNA polymerase is going to stick on, and we are then going to run and create new DNA. This is the extension phase with DNA polymerase. This DNA polymerase is special. If we were using our regular DNA polymerase for me and you, this wouldn't tolerate this kind of heat. It is at 72 degrees Celsius, which is twice as hot as what it should be in the human body. We're at like 34, 37, something like that Celsius. So this is much, much hotter than regular body temperature, and our enzymes wouldn't make it. But Taq polymerase is an enzyme that was found in the Thermus aquaticus bacteria that the guy found in Yellowstone back in the 1960s. And now we are using that enzyme because it can tolerate the heat, because it grew and evolved in a hot spring in Yellowstone. This temperature doesn't matter. It's totally fine and happy in that temperature. And so that is the DNA polymerase, the Taq polymerase, that's going to lay down the new DNA. And so we just repeat this process over and over and over. Denature, anneal the primers, extend, and make new copies. And you know, the cycle goes on and on. So PCR is a DNA copying machine, which is really cool. We can also use this for RNA. If you are getting COVID tested, you may take a rapid test and get results back relatively soon, but the more secure method is to do a PCR, is to see if you have the viral cells in your sample that you had and they will run a PCR to determine, do you have COVID genome on you? Are we finding the COVID genome? So that is the PCR. We are making copies. We are looking for these specific things. That is the PCR machine. Cloning. Cloning is making copies, basically. Cloning comes in two types. We can make a copy of a little bitty piece. That's molecular. So we are just looking, again, if we were looking to make a little bitty copy of something, we could modify a bacteria and have it copy, because bacteria, we said, can multiply real fast. E. coli can double itself in 20 minutes. So that was the original way for us to copy segments of genes, segments of a chromosome. We would stick that into the plasmid in an E. coli and then have that E. coli breed and breed, and then we'd have lots of copies of this, right? That was molecular cloning back in the day, but now we just use PCR, which is great. Reproductive cloning. Molecular is just a piece, just a little bit, just the DNA, just these little strips of it, just the RNA. When we talk about reproductive cloning, that's an entire organism. It's a whole organism. It's a whole Dolly the sheep. It's a whole black-footed ferret. It's a whole thing for reproductive cloning. So the whole organism is reproductive cloning, while molecular is just going to be, just a strand of DNA, just a little bit there. So how do we mess with that DNA? How do we clone and copy in something that we need to have copied? So with the plasmid project, the whole purpose of the plasmid project is that back in the day, we used to use pig pancreases to produce the insulin for people that were diabetic. And some people were having reactions because it's porcine insulin. It's from a pig. It's not human insulin. And some people were having reactions to that insulin. So what we figured out is that we can then use this method. We can use bacteria to create this stuff for us, which is amazing. But we need to put the human insulin gene into that bacteria. How do we do that? How do we get our human genes into the bacterial cell's genome, that big circular genome that we're talking about? How do we do that? Well, we got to cut it and stick it in there. So when we cut it, we're going to use these things called restriction enzymes. Restriction enzymes. What these bad boys are they are looking for a certain coding. And you in your plasmid project have 8 different ones, 8 different restriction enzymes on the plasmid project that you will take. And I've got HPA2 in my hands here. And so I've got GGCC. I got GGCC for HPA2. And wherever this restriction enzyme comes across on our strip, here's my handy strip, wherever I find this specific coding, if I find 2 G's and 2 C's together, just like this, then this enzyme, this restriction enzyme, HPA2, would cut it. It would cut it. So you would take your restriction enzymes, and you have 8 of them, and you are going to run through your strip and look and see where they would cut. And I want you to mark that with a pencil. Don't cut it yet, just mark it with a pencil. And you're going to do this for all 8 of them. You're going to do this for all 8 restriction enzymes. Some restriction enzymes are going to cut the plasmid. My plasmid is, I printed it off on green paper. You can print your DNA genetic material on white and then highlight it, use crayons, markers, whatever you want to do to designate that one of these strips is for your plasmid and the other color strip is going to be the insulin gene on it. And then you will find both of these things, run your restriction enzyme through and mark all the cuts. Some of these things will cut just the DNA with the insulin gene on it. Some of these things will only cut the plasmid. You are looking for one, there is only one restriction enzyme that will cut the plasmid, mine is green, whatever color you want to do your plasmid paper in, you are going to find one restriction enzyme that cuts this guy once, the plasmid once, plasmid, one cut only, we don't want to cut the plasmid more than once, that would be bad. And then you want the same restriction enzyme to cut this strip, your DNA strip that has the insulin gene in it. You need to cut that above the insulin gene and below the insulin gene. You don't want to cut the insulin gene. That would be terrible because then you're not getting the whole gene and you're not going to code for insulin. You'd only get half of the insulin and it's not insulin. So you need it to cut above the gene and below the gene, but don't cut the gene itself. So 2 cuts on the DNA that has insulin, 2 cuts. That's what we're looking for out of our restriction enzymes. 1 cut on the plasmid and 2 cuts on the DNA strip that has the insulin gene in it. So you put all 10 strips together in order, 1 through 10. You've taped them or stapled them, however you need to do it. And then you are going to look for one restriction enzyme out of those 8 that cuts this strip above the insulin gene and below the insulin gene. And that same restriction enzyme will cut your plasmid strip 1 time. Only one of the 8 does this. All the others, aren't going to do that. So that one thing is the special one. When you find that one restriction enzyme that cuts, just like we said, the only one that works. When you find the one that works, then you're going to actually cut your genetic material. So the way we cut it, we are going to end up with these things, sticky ends. These are sticky ends. Sometimes there's blunt ends. Some restriction enzymes have blunt cuts, but our restriction enzymes have sticky ends. And that just means that they're uneven. They're not straight across. They are uneven, and those sticky ends allow a better bond because you're going to get multiple hydrogen bonds that will seal this a little bit better with our restriction enzyme. So go through those for the plasmid project. Oh, it takes a long time. It does. It takes a long time to run through all of those restriction enzymes, but you can do it, and you will cut only the restriction enzyme that does this. It cuts the plasmid 1 time, and it cuts the DNA with the insulin gene on it twice. And there's only one restriction enzyme that does it. When you finally find the one that works for you, actually cut your paper. Actually cut it, and then you're going to put it together. You're going to pretend like you have ligase, and you're going to stick these things together, and then you're going to end up with one gigantic circle, one really huge, you can't see it because I'm green screening, but I have one really large circle at the end of my plasmid project that I have put together. I put all my sticky ends together and they merge, and it looks really good, and you're going to have one big circle. So that is the goal, is for us to get the insulin gene into that bacterial plasmid. And so then every time that bacteria expresses its genes, it's going to create insulin for us, which is amazing. That's the whole job of our plasmid project. We want to get that in there and make this recombinant plasmid with genes that weren't originally its. We've stuck in the insulin gene, and now it is a recombinant bacteria. So that is what we're doing here. In this case, we have our foreign DNA that for our project, we're going to say is insulin. And we want to stick that stuff into the bacterial plasmid. So we're going to use a restriction site. Whatever restriction enzyme works for you, you're going to cut it at that location, and you will cut them in there. It's going to create sticky ends where it kind of does that little, it's not an H, but it does that lopsided side where we get multiple hydrogen bonds across those nucleotides, and it will make a strong stick. So we've got sticky ends. we're going to put these guys together with ligase and we're going to glue. Remember, ligase glues all the DNA together. So it makes it one pretty strand. So we're going to use ligase and put these guys together. Here's DNA ligase to reattach the DNA, stick them all back together. And then in this case, if we were actually in the lab and performing this, we need to know, did the plasmid take? Is it working correctly? Otherwise, did we waste our time and we cut the gene but it didn't get in? Or we cut the gene, the bacteria is not using it? What's going on? We need to make sure that this is actually working. So here we would take it and test it. So we're going to combine these bacteria. We're going to put it into different we're going to see if these plasmids are going to be taken in by these different bacteria. Are they going to absorb this material and pass it on and multiply? So we are going to put these guys in. This is important to note at this time. This AMP is ampicillin resistance gene. So it's an antibiotic resistant gene. I told y'all that plasmids carry cool properties and these bacteria will swap these back and forth, and you can have this cool thing and I'm going to take this cool thing, and they will give each other things like antibiotic resistant genes. So that's cool when we're going to test it in the lab. So here we are going to put it and grow these guys. We're going to try and culture these guys, and we're going to put them on a plate, and on a plate with ampicillin, on a plate with ampicillin. So if they have an ampicillin resistant gene, they'll be fine and they'll grow on this plate. If they didn't, if something happened to that plasma and it's not working properly, then they're not going to grow. That ampicillin plate would kill off those things. So we know everything that's growing on this plate has at least the ampicillin resistant gene in it, the green section. This is intact if it is growing on the plate. Now, we are going to see another thing is can they metabolize these things? That's what we're looking for. So if they're going to change color when they are exposed to the enzyme produced by this, and if it does not change, it has the foreign insert, they are going to have the recombinant plasmid. So we have white ones that did not change color. These guys have the plasmid because when we know they do because they didn't change color, and the blue ones don't have, they have plasmids without the insert. So they change color if they can metabolize this thing. If they don't, they will stay white. And so we can determine who has the plasmid and who doesn't. So that is what we're looking for. We're looking for a successful growth of the ones that we are looking for, the ones that have the plasmid. So this is a way of molecular cloning. This is your plasmid project right here. At least this portion right here is your plasmid project that you are doing as far as the crafting, paper cutting section of the plasmid project. Later, you're going to have the 6 questions that you'll do in your Word document and the table that you will do in your Word document to fill out that we discussed previously when we talk about translation, transcription, and translation. But this is the crafty section where you get to put it all together. So there's a little bit of fun in everything. So that was molecular cloning. That is the plasmid project. Please let me know if you have questions about the plasmid project. Don't wait until the last second to do the plasmid project. Reproductive cloning is the whole thing. Molecular is just a piece, just one gene, just a little strip of DNA, but reproductive cloning is the whole entire animal. So our first reproductive cloning success was 1996 when I graduated high school. That's how old I am, people. So I graduated high school in 96, and this is when Dolly came to be. How did Dolly come to be? So Dolly came from some mammary cells, and that's the name. If you know Dolly Parton, you know what I'm talking about. So we had an egg donor, and we are going to enucleate or take out the egg nucleus. And remember, eggs were haploids. We're going to take out that haploid nucleus and we're going to put in some stem cells that are mammary stem cells. And that is a diploid because it's regular cells in the body. So it's got both sets, right? It's a diploid nucleus. We're going to take that and we're going to stick it into this. And then they do some sort of current pulse. I couldn't tell you exactly what the mojo magic is. That presto changeo makes that cell start to replicate. So that is the science behind it. You take the donor egg from this sheep and we use the nucleus from this sheep, and the nucleus came from some mammary stem cells, hence the name Dolly. We take that nucleus, we stick it in the egg, presto changeo, we're gonna zap the egg and it starts to undergo cleavage. It's going to start to grow just like a regular old embryo. We're going to take that embryo just like we were in IVF, just like we do with people. We're going to take those embryos at, X number of cells, 16 cells, 32 cells. We're going to take that embryo at that level of cells, and then we're going to implant it. So they took the embryo, they implanted it in this surrogate sheep, and she grew Dolly. She grew little baby Dolly. So Dolly was our very first reproductive clone animal successfully, was Dolly. And now just the other day, I saw this article, I guess maybe yesterday or the day before, about now we are starting to clone. We've been cloning a lot of different things, but we have started to clone endangered species. This was the first time that we cloned native species to the US, endangered species, and it is a black-footed ferret, which is super cute pictures. If you click on the article, super cute. Cute little guys from Wyoming. So that was reproductive cloning. So don't forget the difference between molecular cloning, what we're doing in the plasmid project, recreating the insulin gene so that the bacteria can form insulin for us, and we can use that. Reproductive cloning was making a whole new organism, sticking the genetic material of one thing into an egg, zapping it, and having it grow like a regular old embryo, and plant that embryo into a female and give birth to this new organism. So that is reproductive cloning. If anybody has experienced IVF, you know what I'm talking about, where we have the egg and the sperm, they come together, they fertilize, they grow for a little bit, and then we freeze those embryos, and then later, when you're ready, you will take those embryos and have those implanted in you. There are some more things that I wanted to talk about this. Did I have it in here yet? I don't. We aren't there yet. There are a few more things that I wanted to talk about that's not really in the textbook so much, but I did want to kind of touch on it. Is gene therapy. I pulled these slides from my 1107 class. Gene therapy is another issue that, did we talk about it later on? We didn't. It is where you might have a defective gene for something. Maybe you have some sort of disorder where your genes are not creating the proper expression. They're not doing what they're supposed to. You've got a mutation or something going on and you need to fix that gene. We can fix that certain genes with gene therapy. There are pros and cons to this. Maybe if you are able to get this done, then it fixes the gene and you don't have the situation anymore. But there are other cons to this situation. How does gene therapy work? You are going to just do your molecular like we did before. You're going to modify the genetic material and put in the good gene. You're going to put the good gene in a vector. In this case, the vector is going to be a virus. And so the virus, we know viruses get into cells, right? They take over the cells and they kind of mix their DNA and hijack the cell. So we're going to put the good genes. Here's our new gene, and we spliced it into the viral DNA. And then we're going to use this adenovirus. It's going to get into the cell and it's going to mix its DNA with the cell of the DNA, and in the hopes that that will replace the bad gene, that the good gene will now replace the defective gene, and that is gene therapy. Again, there are pros and cons to it. I'm not going to ask you about this on the test, but I did want to talk about it. There are a lot of ethical concerns about this. Which genes are we talking about? Which ones are we feel like we should modify? Who decides who gets to modify them? Who is responsible for regulating this? Whole bunch of concerns. I mean, a whole can of worms is involved in modifying genomes for things. CRISPR is also not in the textbook. I don't know why it's not in the textbook. It is sort of newish technology, but I definitely think it deserves a spot in the textbook to talk about CRISPR and what it does. I have some videos in here that explain it. This is the Bozeman science guy that I love so much. He's awesome. But CRISPR comes in and it basically can edit. It can like cut in places and stick in what we wanted with this CRISPR. Is pretty cool. It is genetic editing, and there's all sorts of possibilities with this. So I've got some articles here um, coming out of these first two are coming out of China, where they are struggling in their labs to make all sorts of breakthroughs. And this breakthrough was to fight cancer. Can we cut out the genes that maybe potentially cause cancer and stop it? Can we just attack the cells around the tumor with this? And so it won't spread anymore. There's all sorts of potential possibilities with CRISPR. But if you watch Bozeman Science, you can figure out how CRISPR kind of works. You can see possible things that are going on. I want to say this was 2018 or 2017, something like that. This one was where they went and modified human genomes in embryos, which is a big, big no-no. We're not supposed to be experimenting with people, but somebody did. So I've got an article here about the scientist that did it. It was two embryos, so it was twins, and he modified one and he didn't modify the other as an experiment to see what would happen. And you don't do that. You don't do that. But he did. And so that's a article about that and the implications behind that. No one knows how that's going to affect these two girls going forward with the experiment that this person did, and more articles about um, what CRISPR could possibly do for us. So this is really cool stuff that legit in the last couple of years, these articles have been coming out. So CRISPR is neat. You should check it out. Um, Transgenic and other terminology, genetically modified things. So I said that we're not supposed to play around with human genomes. It's a big no-no. It's pretty frowned upon to play around with people, but people are doing it. This was a study here where, when we talk about transgenic is usually taking genetic material from another species and putting it into a different species. You are combining genetic material from a bug and you're going to put it into a plant or whatever. That is a transgenic animal when it has foreign DNA in its little host body. So this study, and this one I want to say is at least 5 years old, is where they were trying to see about can we grow human organs in animals? Can we make a chimera? A chimera that's got human genome and animal genome. And so they did it. These scientists did it. They made a human pig chimera, a human pig chimera. And I don't remember how many cells this thing got to. I don't remember if it was 32 cells of splitting, 100 cells of splitting. I can't remember what stage it was before they ended the experiment, but it was viable at that point. It was a human pig chimera that they were able to successfully create. And it is on hopes that these animals would be able to grow, a human pancreas or a human liver or a human whatever. And then we would harvest that animal and take the liver from it, and it would go into a person instead of having to result on the donor list from a dead person. We could potentially grow these things in animals. that was the purpose behind this. I have this older movie here. It's The Island. It's Scarlett Johansson, um, way before Avengers and all of that business. Is this movie? It's a sci-fi movie, lots of action, but the premise behind this movie, and again, it is science fiction, but it's these two characters, and they're living on this kind of lab where everything is perfect. They're fed this special diet. They exercise every day, and at the end of the day, or maybe it's the end of the week, there's this big party where somebody gets chosen, and they get to go to the island, and at the island. It's supposed to be this wonderful vacation that you get to go and you get to leave the lab and you get to go to the island. And then come to find out that when they go to the island, it's not actually a vacation. They are going to be killed and harvested because it turns out that the people in the lab are actually clones of somebody out in the real world. And so it's about them figuring all this out and escaping out of the lab. So it's kind of an interesting premise behind this movie that is, I want to say early 2000s is when this thing came out. But it is a really cool meaning behind this movie that is all about trying to talk about how we can get organs and things for people. Um, but on that one, it was on the dark end of the spectrum. So transgenic animals have foreign DNA inside of them. If we were talking about GMOs, again, GMOs are genetically modified organisms. Usually that's anything in agriculture. That is my dogs that are sleeping on the bed right here next to me. They are genetically modified because we manipulated the dog traits and bred those. In more recent times, genetically modified is usually talking about um, it's usually an organism that we've modified, a plant that we've modified and messed with their DNA molecularly. We have done something. We've added in a gene. We have stopped a gene from expressing for a GMO. Um, Transgenic, we talked about typical genetics back in the day. We would have a phenotype, what you are seeing. I have dark hair and I have dark eyes. So that's my phenotype. What are the genes that control that? And so we would go from a phenotype to the genotype. How do I find out what genes produce this? And now we have reverse genetics. And so that's some kind of really cool stuff that's been going on recently where if you had the DNA of, say, a suspect at a crime scene. We have DNA from a crime scene of a perpetrator that has done something, but we don't know who this is. We don't have them in any database. We can't find out who they are. They're not registered. They haven't done a 23andMe. They're not in the system, but we do have their DNA. So now we can do reverse genetics. And with reverse genetics, we got your DNA, we can try and figure out what your phenotype is. So now they can do this. I need to find an article and stick it in the PowerPoint, but I can't remember what it's called, but they will take the genes of a sample of a suspect and try and figure out, they can code that in and get an approximation of what somebody would look like based on their genetic makeup, which is super cool that you can create this, virtual mugshot of somebody based on just the DNA left behind. So that's reverse genetics where we have the genes and now we're looking for the phenotype. So super cool stuff that is going on in the world in the last couple years. It's cool. This is biotechnology. This is one of my favorite chapters. I think it's really neat to talk about. So next one, we're going to talk about medicine and animals, genetic testing and stuff. So that is going to be the next video in just a little bit.